My name is Beraut. I'm a fourth-year undergrad at UCLA, and today I'll be talking about biomonitoring with environmental DNA. So this will be a series of summarized findings where each project kind of inspired the next one. So molecular techniques have opened up a lot of plant microbe questions that we can explore to further our understanding of molecular ecology. And microbiomes in the soil have been seen to be associated with their neighboring plants in the rhizosphere. And they've been seen to be dynamic in their taxonomical composition as well as their function. And so this study on the left here by Mead and al. 2016 shows that microbe species richness varies with tree age, but that microbial ecological traits are conserved. And so this study in the middle here by Martinez et al. 2017 was interested in looking at fire disturbance on home oaks in the Mediterranean. And they found that undisturbed and burned oak forests exhibit differences in the microbial relative abundance of the dominant phyla, that the composition remained generally unchanged. And they were able to tie this to tree fitness by identifying specific genera that were more abundant in the burned forest that helped oaks grow. And this was most clearly seen in the genus Archrobacter. So what about the rest of the biome? So these people have found some pretty interesting relationships between bacteria and trees, but there aren't a lot of studies that have used these same methods to look at protists, algae, invertebrates, or fungi. So I plan to explore that as I go through. So what is environmental DNA? So this picture kind of represents what environmental DNA really is. And as you see, there's a lot of organisms, plants, fungi. And here we have a citizen scientist taking a soil sample. And so what you can do is you can get a fingerprint of all of the organisms that shed DNA in this region from your environmental DNA samples. So at UCLA, we're spearheading this citizen science program that is trying to capture the biodiversity across the entire state of California. So how do we do that? So we take biological replicates, we extract the DNA, and then we use a series of primers to target these 5 barcoding loci: bacteria, eukaryota, metazoa, fungi, and plants. We then sequence these and process them through these software pipelines to assign them to OTUs. Then we do our community ecology analyses, and these analyses will eventually cycle back to the citizens that took these samples in the first place. So I will be presenting a series of studies that I've done and worked on that incorporate these same methods to try and analyze these microbiomes. So the first one that I looked at is eDNA in oak trees. So this was a project spearheaded by my mentors at UCLA, Meyer, Victoria Sork, and the Sork lab, where they took oak trees of different parentages and provenances and grew them together in a common garden. And we were interested in seeing if there are any differences in the associated microbiomes. And what we see is that species richness actually varies by a few variables. We see that the species richness in Curomycota, Cercozoa, and Ascomycota varies by location, and that Heterocomps and Basidiomycota vary in species richness by being under the oak trees. And then based on the type of oak tree you find them underneath, we see that species richness varies again in the Heterocomps, Basidiomycota, and Ascomycota. So these findings indicate that the plant, the presence and genotype, as well as the location, both regulate the biomes around them. So I was interested in looking at how things like community disturbance could affect these communities, and I was most interested in seeing how fire could do it given California's current state. So fire can cause a lot of changes in the region with geochemical changes in the air affecting all the plants. As the plants die, there's a lot of erosion and a lot of water redistribution that causes flooding. And so they can cause really drastic changes to the landscape. So I was interested in seeing how the plant and microbe communities change. So that's when I started this pilot study looking at environmental DNA and fire in the Klamath Mountains. So what we see on this map here, in this region where the samples were taken, this is a map of Cali DNA results from all over the state used in a gradient forest model in biodiversity, and we see that in that Klamath region specifically, there's really high community turnover with the swirling pink and green. So this is a really interesting area to conduct this study. So how we did this study was sampling. In one summer, we took 18 samples over 4 different fire sites, and you can see up there the years where these fires burned, as well as adjacent unburned areas, and we grouped them together for the sake of these analyses. And we took metadata with all these samples, including things like burn intensity. So now for some results. So this is looking at species richness for the 5 barcoding loci I talked about earlier, the bacterial, eukaryotic, invertebrate, plant, and fungal communities. And we're looking at species richness over fire site. And the only significant effect we saw was an increase in species richness for one of the fungal fire sites, actually the oldest fire site, compared to the unburned sites. what I was more interested in was trying to find if fire changes community composition. So these are principal coordinate analyses based on a Jaccard distance matrix, and we see the points here are colored by fire site. And coloring the points by fire site, we don't actually see very good sample clustering. But when you look at these boxes that I overlaid, comparing the areas of the unburnt samples to the burnt samples, we actually see that the unburnt samples occupy a different set of pink boxes are unburnt. They occupy a different area on the ordination plots compared to the burnt samples. So this is present in the bacterial community, the eukaryotic community, and the fungal communities. But as we move to the plant community and the invertebrate community, these patterns all but disappear. And so I was interested in trying to find out more, so those give a good qualitative assessment of community composition changes. We're also interested in doing a quantitative assessment based on the metadata collected with our samples. So here we see that fire site is a significant driver of variation for the bacterial community, eukaryotic community, and the fungal community. And this is in line with what we saw with that gradient forest model of really high community turnover with the swirling patterns. But the plant community is a little different. The only significant driver of variation for the plant community was burn severity. And so these are some interesting patterns to see, but when we're looking at these patterns, we're looking at all of the taxa present in the data set, which is a lot. And so it's hard to kind of find what are the really interesting interactions. And so it raises a few questions. If only some taxa are important for plant fitness in disturbed environments, what are they? And are beneficial organisms associated with plants that do well in these burned environments? So the next step to try and learn more about these was to create this ecological community network. So this big messy hairball represents over 13,000 taxa interactions between the over 700 taxa we had in the data set. And you can see that there's 5 vascular plant genera and 8 bryophyte genera that consist of these green dots scattered throughout. And there's a lot of cool interactions that have been found in here, but with so many in this big hairball, it's kind of hard to find what are the interesting ones. So what we can do is we can create a simpler version. So this is looking at the fungal community and the plant community interactions. And what I've done is I've highlighted what I find to be one of the more interesting interactions in this data set. So what we're looking at here is Coauthors thistlephoris and rhizopogon. So Coauthors is a shrub that has been known to do really well in post-fire sites where there's been a high severity burn. And rhizopogon, in a completely separate study, has been shown to increase in relative abundance in the years following the fire. And so it was really interesting for me to be able to see that these 2 fire-responsive taxa are associated in our network. And so with that big hairball and those 13,000 interactions, I wanted to find a way to make these public. So that's when we reached out to the people at Globi. So Globi is an open source repository containing millions of interactions, but we currently have a really big hole in these plant microbe interactions. And the reason is that no one has tried to upload these eDNA interactions before. So we reached out to the people at Globi, and they were quite excited about it. And so I am now in the process of submitting a manuscript to APPS that outlines a workflow, so anybody who has an eDNA dataset can publish the entire interaction network online to these repositories. And I chose the relational ontology term ecologically co-occurs with to describe these interactions. So I learned a lot about assessing those fire-prone systems with that pilot study that I did, and to try to understand how these disturbed systems can recover moving to the future, we can look to the past. So this is a project that was spearheaded by a colleague of mine, Martinez at UCLA, where we're looking at Lake Elsinore charcoal concentrations through a sediment core. So this sediment core, the layers down at the bottom date back to the late Pleistocene and go through the Holocene. And as we move across this timeline, what we see around 12,000 years ago is this huge spike in charcoal concentrations that lasts for a few hundred years. And it's so drastic that I kind of imagine this is what Lake Elsinore looks like at the time. Just an artistic representation. But what's really cool is that a different study done on the pollen counts of that same time period, so these line up on the same time period, there's actually a large increase in quercus pollen and a decrease in the pine pollen over this time frame. And so looking at those those pollen charcoal associations, I was interested in seeing how my DNA analysis could find interesting stories to tell through that timeline. So I only have some preliminary results to show, but what we're looking at here is a subset of 5 samples. So the sample in orange represents sediment layers from about 100 years ago, going down to the dark green, representing sediment layers as old as 30,000 years ago. And on the x-axis here, we're looking at the relative abundance of reads from the environmental DNA samples, and on the y-axis, this is just a little snippet of all of the plant taxa that we found in this data set. And what you can see up here at the top is with these large amounts of dark green and regular green. Oh, another thing is that this light green, this 11,000 years ago, was taken right after that peak in charcoal concentration. So we can really look at this and try and see how the relative abundance changes or along with this fire history. And so looking at the relative abundances, we see this dark green and green a lot at the top, and then the light green consists of a smaller region, which means that these taxa decrease, these orders decrease in relative abundance after the fire. But as we move down to the bottom, we see really high portions of the bar being made up of that light green color, and so these plant orders at the bottom increase in relative abundance after the fire. So some conclusions. From that oak common garden experiment, we learned that plants and their locations regulate the biomes around them. And then from that pilot study looking at fire in the Klamath Mountains, we learned that communities largely had different composition by site, except for plants, where significant variation is driven by burn severity. And then through that network analysis, I was able to find some cool interactions, such that some plants that do well in post-fire sites are associated with fire-responsive microbes. And then what's the most exciting thing for me from this whole study is that anybody with an eDNA dataset can publish thousands of novel plant, microbe, animal, and other interactions from these environmental DNA datasets to biotic interaction repositories. So I'd like to thank a lot of people who helped me with this, the Sork Lab, Bob Wayne, Glenn McDonald, Mishi Lin, and Christopher Adlam. And CalDNA is a very citizen science program, so if anybody is interested in getting an eDNA data set of their own and doing some of these analyses, please check out the website. Thank you.